Hello and welcome to Inside Exercise. I'm Emeritus Professor Glenn McConnell from Victoria University in Australia, and I'm also currently a Danish Diabetes and Endocrine Academy visiting professor at the University of Copenhagen in Denmark. The idea behind Inside Exercise is to bring to you the absolute who's who of exercise research, so exercise physiology, exercise metabolism, and exercise and health. And what I really want is for you to get your exercise research information from the research experts rather than from influencers. And indeed, today I bring to you Professor Wim Derave from Ghent University in Belgium. He's an expert on muscle, specifically muscle fibre types and muscle adaptations to exercise. He's doing some really interesting studies that are challenging our prevailing view about muscle fibre types. So we tend to think of muscle fibres in humans as being of three types. So type 1, which is slow twitch, so slow oxidative, which is good for aerobic metabolism. And then the fast twitch fiber type 2A, which is fast oxidative glycolytic, so has aerobic and sort of anaerobic sprint type performance. And then type 2X, which is your fast glycolytic, which is not very good at aerobic metabolism at all, produces high forces, and is very good at producing lactic acid. Now, in very interesting findings, Wim is showing that most people have no 2Xs. So no 2X muscle fibers at all. So this is very interesting. Wim makes the good point as well that we don't really use muscle fiber type enough. Wim talks about how in the 1970s and 80s, people like David Costal and Bengt Saltine, who are legends in the field of exercise physiology, were using muscle fiber type for talent identification, etc. But he makes the point that we haven't really progressed on from that, and there's very little use of muscle fiber type. So as he says, there's talent identification, but also he's got evidence that the likelihood of having hamstring strains is affected by your muscle fiber type, that you could potentially use it for tapering and perhaps overreaching. So he's saying a lot more work needs to be done to use muscle fiber types more optimally. He also talks about non-invasive ways of estimating muscle fiber type. And then towards the end of the chat, we talked about something very different, which I was totally unaware of, which is the fact that antihistamines may reduce your responsiveness to training. So if you take antihistamines, you may not adapt as much. I found it really interesting. I think you will too, so stick around. Now, what I wanted to point out to people is that you'll see in the notes, so whether you're on YouTube, Apple Podcasts, or Spotify, or whatever, that I have description of all the different things we talked about. And next to that, I have the time that we spoke about it. So obviously, I encourage you to listen to the whole thing. But if you wanted to, you could skip around a little bit. What I tend to do is start off with the basics, and then we get more complicated as we go. So, if you don't want to hear the basics, you can look at the timestamps and go straight through. So if you're on YouTube, you can click on the time. It will take you to that point. But on Spotify, Apple Podcasts, etc., the timestamps are written there, but you just have to take note of them and then fast forward. You can't click on them. The other thing is, please do me a favor by liking, subscribing, also leaving some comments if you're on YouTube. That all helps to get the message out. Hi, Wim. How are you? Welcome to Inside Exercise. How are you doing?
Fine, thanks. And by yourself?
Yeah, good.
It's great to be on this series of talks. I really look forward to it because it's a very prestigious lineup of all kind of scientists I have looked up to and admired over all the years. So it's I think it's a great initiative. Thanks.
Oh, thank you very much. I think you're the fourth one in a row. Talking of prestigious scientists, so we had Eric Richter, who was your PhD supervisor, on a couple of weeks ago.
Yes.
And I think you're the fourth one in a row that's had a link to the University of Copenhagen, but we'll probably get to that soon. All right. So what I like to do at the start is get a bit of a background on you and ask, were you like a scientist who then took an interest in exercise, or were you quite often, I think probably on average, most of the people I've had on were sort of sports types, exercise types who then thought, oh, I can actually study this. What was it like for you?
Well, I was a sports person, but not in a high performance level in any way. Mm-hmm. I was a bit of a generalist, I would say. But the thing is that very early in my sports science studies, I really got intrigued by physiology, and already during my students period in my third year, I I had an Erasmus exchange to Copenhagen. So then I first got to, was Jens Bangsbo, who was my teacher for exercise physiology and so on. So I was intrigued even more. And then, after I graduated, I started in a PhD, and I immediately knew that I had to go back to Copenhagen to do some real science. And so I ended up with Eric Richter, it was a colleague from Leuven, Peter Hespel, who advised me, who was also been a postdoc with Eric, to go with him. I really think that this has been one of the most defining moments for my own career.
Yes. So some people that would have seen, I've mentioned that I'm actually doing my 4th research visit with Eric. So he's great. And the methods you can do and things are amazing. All right. So we're going to talk about muscles. So we've had some talks about blood flow the last couple. We had Ulvia Hanston two times ago talking about blood flow and muscle. And we had VO2 max with Jose Calbert. And, capillarization and fiber type and things got mentioned here and there, but not really, thinking more, I guess, about sort of central, like blood delivery and things, but not really thinking about the fiber types. So today we'll be talking about the importance of fiber types, and we'll get into some really interesting stuff where you've been finding, for example, that there may only be, realistically only 2 types of muscle fiber in humans rather than
humans.
Pardon?
Yeah. In humans, yeah.
In humans, exactly. Compared to other species, and we'll talk about all that. But why don't we just start off, before going too hard, to give a bit of a background. So a lot of people would have heard of, that person's fast, they're probably fast twitch, and that person's endurance, I think they're probably slow twitch. Why don't you just tell us what are the different fibers in humans, and what do they do, and why is it important?
Yeah. So we scientists and even non-scientists have always seen that there's different types of meat or muscles. So there's red and white muscles that you can find like in chicken or in doves or whatever. So we've always seen that there's different types of muscle. It seems that within the organism, this also can differ, like the muscles that are important for posture, for balance keeping, then have to be active for hours and hours a day, up to 8 hours or more. They are also from this wrapped muscle or slow twitch muscle, and then you have this more burst-like activity muscles who can produce much more power, but they are also fast fatigable. And this is the white muscle. So we've had this in the animal kingdom, and we clearly saw this also in the humans. Not so much the color, but when you have specific techniques to stain muscle fibers, we could easily see that there was a mixture, a mosaic of white and red fibers, or the fast twitch and the slow twitch fibers.
Yeah, that's very interesting. I often use the example of you mentioned the chicken. So chicken walking around all day, their thigh muscles are more sort of red and even more oily. So you may talk about how there's more intramuscular fat and things, and those sort of red, slow, less fatigable type muscles. And then then if they get a fright or something, they fly for about 5 seconds and then drop to the ground. And that's the more the breast muscle, which is very white, very dry, not very fatty. And you're saying that's the fast twitch. They can produce a lot of force, a lot of power, but they fatigue quickly.
Yeah. Yeah, and you also see that between species. So you have this entire species that are fast twitch and entire species that are slow twitch. So that is, we see that this specialization towards speed, for example, in a cheetah, which can produce fantastic speeds for a couple of seconds only, and then these other animals like migrating birds who can produce the same force for days without fatiguing. So that is really something that has been observed consistently in many species.
Yeah, and why is it that because I guess we're going to talk about how humans tend to be 50-50, but it can depend, like sprinters will be more fast twitch and endurance to be more slow twitch. Why is it that the species differences? Obviously, okay, cheetah has to be fast, as you said, migrating bird has to be slow. But why is it that we have more sort of 50-50? How's that worked out, do you think?
Mm-hmm. Yeah, we have 50-50, but actually we are slow. We are slow twitch endurance species. There's almost no other land animals that have so many fast fibers. Even if we would have about, on average, 55% of these slow twitch fibers. All the other big apes would have 70-80% fast fibers, and if you go to antelopes and all the big cats and so on, they have like 95% fast fibers and so on. So we are really a slow species, or an endurance species, and that is something that I think most people don't really realize. But we kind of, in course of evolution, we have decided almost to make a strength or a talent, our endurance a talent, and it's even more than a talent; it's a weapon, because we hunt down our prey even before we had weapons and we were hunter-gatherers. We just ran down our prey for hours and hours, especially in a hot climate as in Africa, where life for us started. We can run down every animal from the savannah as long as we keep doing it for hours and hours. The fact that we don't have hair or fur, or it's all part of this machine or this fantastic weapon we have, that is endurance. So we as a species should really look at ourselves as a slow twitch specialist and an endurance type of animal.
Okay, I'd heard that. I remember they were talking about with the Australian Aborigines, they could chase. Maybe this is wrong. The thing I'd heard was that they could chase the animals, and then the next day they'd be like sore. The animals would have muscle soreness, and then they could grab them. But you're saying you would actually wear them down over hours. So the muscles, as you said, most animals have quite a lot of fast fibers, so they tend to fatigue. And we would wear them down and then grab them or stab them.
Yeah, pick them up. Pick them up. They are completely exhausted and fatigued. It's probably a combination of fatigue and heating. So we have more sweat glands per square millimeter, and we have a very good system to sweat and so on. So where other animals would have to take a rest in the shadow and cool down, we can keep on doing that. So this is really our strength. We can run marathons in the heat, and if you would do an Olympic Games and all animals can take part, and you do it in Nairobi, for example, we would win only one discipline. That is the marathon. We are lousy in speed, and when you're lousy in something, you can as well forget about it. Like in school, you're good in maths and not in languages. Then skip the languages and completely specialize on the maths. That's what we've done. We realized that we are really bad in speed. We can't even catch a single animal by hand. I mean, we cannot even catch a rat or a mouse. We are super slow. And then we said, okay, let's specialize in this slow twitch thing, this endurance thing. This is really what we are. This is our middle name. And that is something that, to me, is also the reason why we are losing the super fast fibers. We are dropping that part, but we can go into that later. Yeah,
we will. We can't help talking about the marathon and mention, I don't know if you saw, Calvin Kiptum ran 2 hours, 0 minutes and 35 seconds yesterday in Chicago.
And then 2 weeks ago in the women's marathon. It's really amazing. That's probably one of the most typical activities for Homo sapiens to do. All the rest is a little bit artificial and so on. But I really like that the marathon is the last event in the Olympic Games, and that's the one where the medal is given at the closing ceremony. It's the most important discipline in sports. It's what we are as a species, is this endurance running. That's really how we define ourselves. And if we ever skip our name because we call ourselves Homo sapiens, but I'm not sure with all things that are happening in the world whether we're so smart. So if you want to drop the sapiens name, maybe we can bring something in about running or marathon or whatever, because that is that really defines us as a species. Yeah.
So what about, we have to survive and reproduce. So survival can be wearing down an animal to get the food. Yeah. But what about getting away from prey? Surely that's important. So, sprinting to a tree and building up your lactic acid and then recovering in the tree and hoping the cheetah can't climb up and whatever. You're saying we're not too good at that, and we just didn't evolve that way. We used our brains to.
Yeah. I mean, there was this story a while ago from it was on the BBC News website of a Kenyan village where there was cheetahs actually messing around with the goats, taking goats from this farming village in Kenya. And they were fed up with that, and they said, well, but let's run after these cheetahs, which makes no sense, of course. They are so fast. But what they did is the whole village and children and everyone with just by being in a majority, they made the cheetahs go away, and they started running after them. And after a few kilometers, they could just pick them up from complete exhaustion. So that is really what we still do. Not so much and not so often, of course. But this persistence running or persistence hunting has been something that is done up to today. There's even a documentary of BBC Earth of David Attenborough where he explains this persistence hunting that's still happening in the San people in the south of Africa. So it's still actual, even in groups that don't have weapons or whatever. You can still run down animals quite easily. So slow twitch is our solution to.
Okay. And as you said, we'll talk about the intricacies about what sort of fast twitch fibers we have. But if we've just been up till now, we've been talking about slow versus fast. How much of that is are you born with, and how much does it change? Because I mean, along the way, there was a lot of talk about how it can't really change. Whatever you're born with, that's pretty much it. And my thinking, I guess, was always that you could change it maybe a little bit, so but not massively. If we talk about slow versus fast, and then we'll talk about the 2A and the 2Xs later, how much of the endurance, so Calvin Kiptum, if he's running 2 hours and 35 seconds and we biopsied him and find he's 80% slow twitch, would he have been born that way, or would he have been maybe 65 and then a little bit changed to 80? Or
yeah, there's no consensus really. It's quite difficult to prove that. Because what you do is you have to take a biopsy, and that's a very small piece of muscle. It's 100 milligram or 200 milligram. That's a couple of hundreds, maybe 1,000 fibers of, for example, a quadriceps or vastus lateralis. And that vastus lateralis would have like 500,000 or 1 million fibers. So you take a very small piece of that, and there you count the number of fast and slow fibers. And then a couple of weeks of training later, you do that again. But then you have to take a different part. And then just by random methodological variation, that can be completely different. So we've done studies where we did 4 biopsies in one person. And then you can really see that there's quite a big variability. It can change 10, 20, 30% between one biopsy and the other. So that makes it really difficult to study this question, can fiber type change? And for that reason, probably there's still no consensus. There's still no agreement on whether it can change. What we know is that, for example, the twin studies indicate that monozygotic twins, so identical twins, they have muscle fiber type composition that is much more alike than dizygotic twins or non-identical twins. So we know that heritability is playing an important or really important factor here. That said, I think there's some cross-sectional data that indicate that it's probably can change a bit, like you say, maybe a 10-20% can change. For example, when we look at the upper body of swimmers, muscles in the upper body, deltoids and so on, they seem to have much more slow fibers than an average person, and that is very difficult to explain that would all be because we don't see that in the leg. So it seems to be something that is adapted along the way. So I think there's a little bit of a shift that can be done, but you cannot move from one end of the spectrum to the other. So you cannot make a race horse from a farming horse and the other way around. And so you have a little bit room for change probably, but not so much.
The other thing is, I guess it's making me think a little bit because you were saying the variability between biopsies. But I remember when I did my master's with David Costal, he did one study where he did like 8 biopsies, and he found that, if you just did one, that was very variable, and then if you did 2, it dropped off, and then it became slightly better. So if you assume that 8 biopsies was correct, the percentage you got, then the biggest drop was from 1 to 2, and then it was dropped off a bit. But as you say, most people probably just do 1 biopsy before training, 1 after training, etc. But I guess at one stage, David Costal said he thought if you had 65% slow twitch fibers, then you could pretty much do whatever you needed to do. You didn't have to be like, basically he was looking at all these marathon runners way back, and he wasn't finding that, the ones that were better had 80% than 65, and then if you've got 65%, you won't be as good as someone who's got 80%. He kind of felt like if you had 65%, you could probably pretty much do whatever you needed to do. You didn't have to have like, I've got 68%, I'm going to be quicker than you. You know what I mean?
Mm-hmm. Yeah, that we also saw that in our study where we did non-invasive measurement of muscle topology. We saw that international level or like national level endurance runners, we saw like the same muscle topology, but for sprinters we did see. So there seems to be no limit. The more fast fibers you can get, the faster you will become. But whereas for endurance running, it's not really the case. But if you see Mo Farah, for example, he runs 400 meter in, I don't know, 50 seconds or so on. So you cannot do that on pure slow fiber. So the whole thing about this endurance running is also, it's become a really fast event in the end. And you can oxidatively train your fast fibers, so you can make them have a lot of mitochondria. So it's true, and we also saw that in our data that the more the better only counts for the fast end of the spectrum.
Okay, so with the sprinters, the more you have, the better. But with the endurance, if you've got at least 65, I just pulled out of that shouldn't be much different. But as you said, another point was that your fast fibers can become more aerobic. Now, I guess we're starting to get to the point where we need to talk about the types of fast fibers, because if you're talking about the 2X, which are the fast, that's probably not as much of the case as the 2As. So why don't you tell us about this? So yeah, we've always been, and I was saying to you before we started, that when I learned it was always you had type 1, you had type 2A, and you had type 2B, and then it became 2X. Why don't you just tell us, I don't know, the difference between 2B and 2X, I guess, to start with, and then talk about, what are the balances of type 2 fibers and how you think there's really only probably 2 types in humans.
Mm-hmm. Yeah, so the first studies in the 60s and 70s of last century, a lot was done in animals, in rats, and also in cats. So if you look at the motor unit, so you have a motor neuron with a lot of fibers all attached to it, which are fired by this motor neuron. Then you have this motor neuron in 3 different types. It's called slow, then fast fatigue resistant, so fast resistant, so to say, and fast fatigable. And then if we look at the muscle fibers, we saw the same. It's like slow, and then we call it fast oxidative glycolytic, and we call it fast glycolytic. But that is in animals. And for some reason, we have always thought that must probably be the same in humans as well. And we started staining fibers based on a method which is called ATPase staining, where there's an enzyme which is labeled for acid or base environment, and by like destroying this enzyme in an acid or in a base, we could see the difference between fibers. And there was, if I look at these pictures, I always think it's 50 shades of gray. It's a continuum of a bit of everything. But then after a while, the technique became more molecular. And what we looked at was myosin heavy chain. Myosin heavy chain is one part of the muscle fiber or the sarcomere that actually produces force. It's like a power stroke that is produced by this head of the myosin molecule. And we could see that this comes in different versions. And actually in rodents, we saw the scientists saw that there was 4 types. It was type 1, type 2A, type 2B, and then there was another one, type 2X. And it appeared to be in between type 2A and type 2B. So that is how it works in rodents. And then we checked what is actually present in humans. And there we saw that type 1 and type 2A myosin was there, but type 2X was there, and type 2B was not there. So we still have that gene, but we don't express it. So the 4 basic myosin-based fiber types that are in rodents, we only had 3, and we had to rename type 2B to X because the B was already given to another protein, which we in the end didn't have.
Okay, can I just, I guess, summarize that? Yeah. So, okay, so we're all good with type 1, yeah, and which is the oxidative, which is the slow one, yeah. But then you've got your 2A, because we were sort of changing, we were switching around a bit terminology. So 2A, I was saying was what you were saying was the fast oxidative. So it's fast, it can produce a lot of power, but it also has quite a lot of mitochondria. It can use aerobic metabolism. And then you've got the 2B, yeah, which is a very quick contraction. And you're saying that the mice and rats have that. That's your fast glycolytic. Yeah.
Mm-hmm. Yeah.
But we don't really have that. We have one which is 2X, which the mice and rats have as well, but it's not as fast, not as glycolytic. Yeah.
Yeah. So we don't have the super fast. We have the medium fast and the normal fast, so to say. So we don't have the super fast anyway. So this already shows us that we are a bit moving away from speed as an important thing for our evolution. So to say.
Sorry.
Yeah.
Were you saying that so the 2B, which is the super fast, which is in the rodents, did you say we actually have the gene for that, but we don't express it? Because that's pretty interesting.
That's correct. That's correct. And my point is that also for the type 2X, we have the gene, and we sometimes express it a little bit, but we are getting rid of it also. So we're moving away we're dropping the super fast fiber types. We lost the type 2B, and now we're also losing the type 2X. Because if you do physical activity, you actually you suppress the expression of type 2X. And in a physically active person, usually we don't see any type 2X. So the way to get rid of this super fast fiber type, the way we do it now is simply to suppress it by physical activity. And we know that in the past 100,000 years, hunter-gatherers have always been active. So always suppressing the type 2X fiber type, which means that for the past 100,000 years, we have survived with only 2 fiber types because we were suppressing the third one. Now, sometimes we see in biopsies that there is type 2X expression, but that is mainly in people that have a low activity level, or, for example, spinal cord, which is an extremely low activity level, which means complete paralysis. So we fail to suppress the super fast fiber type if we are too sedentary or sick. But in a normal functioning human body, which is physically active, hunter-gatherers, farmers, and so on, we've probably completely suppressed that fiber type. And even there's one of the most typical genes of these super fast fibers is actinin 3. 20% of world's population already have a knockout of actinin 3. So I think we're in the process of losing it completely. And if we would talk to each other in 100,000 years, maybe we would no longer recall that there was an intermediate period where there was some type 2X expression now and then.
So you're saying it's only really sedentary people, or the people, or as you said, spinal cord injury people, that actually have the 2X fiber. And we're meant to be hunters and gatherers. We're meant to be physically active. Even doing your 10,000 steps, you would not have that fiber. So you would only have the slow, so type 1, and the 2A, which is your fast oxidative glycolytic. So that becomes really interesting because it's hard. It's just like a total flip, right? Because I've always, I guess, or for me anyway, I've just always thought the pure sprinters are going to have the 2Xs because that's, they don't want to have fast oxidative. They just want to have fast glycolytic. Yeah. But they're actually losing it. And it's interesting you say that because I've done some of that myosin ATPase staining. So with the acid, where some people may be familiar with this, where if you look at a cross section of a muscle, you see there'll be all these white ones and some black ones. The black will be the slow and the white will be the fast. And then, as you say, I guess it's shades of gray. But the ones that are intermediate, we'd always call we used to call them 2Bs, but 2Xs, yeah. And then you'd find if you exercise train people, you wouldn't even find those anymore, hardly. They'd be gone. So the idea was that 2Xs would get a 2A. And that's what you find.
There is, not everyone will agree with me. But there's a bit of controversy in the literature on that. What we know is that the physical activity and training suppresses the expression of type 2X. That has been, repeatedly shown. If you do cross-sectional studies on athletes, you will not find type 2X fibers in endurance type athletes. And usually you also not find type 2X fibers in well-trained sprint or power athletes. There's been studies on elite weightlifters, world-class weightlifters. They have 70% fast fibers, but none of those is type 2X pure. So there is only one exception in the literature, and it's mentioned a lot. It's from the Trapeze group, showing that a biopsy of gall injection had 30% type 2X fibers, which inarguably is a top talent, of course. So I don't know it's a case report, of course, but most studies would show that well-trained sprint and power athletes will not have this high number or will have zero type 2X fibers. So I don't know how to place this analysis, and that brings me to another idea. It's very difficult sometimes, or there's problems in how to define a type 2X fiber or a pure type 2X fiber, so to say, because the techniques that we use are usually semi-quantitative or just qualitative. So what we've done in a more recent study now with proteomics, in collaboration with a group in Copenhagen from Atul Deshmukh, proteomics allows to do a quantitative comparison of how many 2A and 2X myosin you have. And there in the biopsies, we could also see no fibers that have, in a quantitative measure, more type 2X than type 2A protein. So that is probably the reason why there's a little bit of controversy in the literature. How do you define a type 2X fiber and a pure or a hybrid or whatever? That makes it a bit complex.
So the proteomics, just so people know, it's a lot of omics nowadays. There's ways to measure all sorts of different changes in muscle, for example. So with proteomics, you'll basically look at all the proteins in the muscle. Now, are you saying that they're doing that on single fibers or pooled fibers or the whole muscle?
Yeah, so we do that on single fibers. So we have a study now where we collaborated with the group of Atul Deshmukh, and we developed or optimized a method for single fiber transcriptomics, and they at the same time in the same period developed a technique for human muscle fiber or single muscle fiber, so not pooled, pure single fiber.
It's one fiber.
Yes, it's one fragment of a fiber. It's a few millimeters long, and it's 50 micrometers thick, and do the whole transcriptome or the whole proteome, which means measuring all the mRNA that's there and all the proteins that in just one fragment. So we optimized this to do it on a high throughput way, and we did it on 1,000 fibers for mRNA, and they did it on 1,000 fibers for proteome. And we came to the same conclusion that that type 2X fibers don't really classify as a separate fiber type when you look at all the genes or all the proteins.
Oh, okay. Now, when you were in your tweet and in your paper, and also, is it Kevin Murak? How do you say it? Kevin?
Murak, I don't know.
Yeah. He also sent a tweet in response to me saying I was having you on, saying that they also find very few pure 2X fibers. It made me think, remember way back, like 20 years ago, I think it was like Eva Blomstrad, I think it was looking at coexistence of different fiber types basically within each fiber. Yeah, is that right? So saying that when you look at a fiber, it's not, some will be just pure type 1, some will be pure type 2A, for example, and some will be like a hybrid. And she went through and looked at them all. Is that still considered to be the case? So that within the one fiber, you can have more than one type of this myosin heavy chain?
Yeah, there are some indications that along the length of the fiber that it can maybe change. This will probably not be the main reason why there is this co-expression that we sometimes have. So we have like pure fibers. We see that on the proteomics and the transcriptomics, and then we see some what you would call intermediate or hybrid fibers who are a little bit on the crossroads of the different types. And it could be indeed that this is because the fiber type changes along the length of the fiber, or it could just be that there's a combination of proteins that are expressed. What we also did is look at not only myosin heavy chain, but also, for example, you could do a fiber typing on troponin T, or troponin C, or on circa, or on tropomyosin, and because they all have a fast isoform and a slow isoform. Okay. And what we see is if we have a fiber which is fast for myosin heavy chain, usually also fast for tropomyosin and tropomyosin light chain and so on. So it makes sense that, I mean, it's like with a car, if you want a fast car, make all the elements of your fast car fast, and just not have one wheel of a slow car on there. So you want the whole machine to be consistent, but still we see that about 20 to 30% of the fibers have deviation from that. They may have a fast myosin, a fast troponin, and so on, but then, for example, a slow circa. So I think there's still a lot to discover there on how this actually is functioning with slow versus fast machinery in a muscle fiber.
Wow. Okay, so that's interesting. And so you're actually saying the coexistence. So with Eva Blomstrad's paper, it would be like she'd take a biopsy, and you're saying if she sort of had, could be like one end of different ends of the same fiber, could be, you know, of the same fiber, you could have a bit slow, it could come up slow one end and 2A the other end, so you get this coexistence. That's hard to kind of imagine.
Yeah, it doesn't make any sense, because you want the contracting fiber to be consistent, right? You want to don't want to be. So we don't really know what is there, but it has also been shown quite recently in rodents, where you can actually see these regions with different expression level. So that is something we'll have to iron out in the next couple of years, what actually is going on there.
Hey, I was just thinking, you sent me, you've got this resource that you make available to people on fiber types, and we'll get to it a bit later on about how you can apply some of this understanding. And I saw you had the sloth. So, if someone's lazy, you call them a sloth, yeah? Because they do tend to just sort of sloth up there in the tree, don't do a whole lot, a bit like the Australian koalas, I guess. And they were like 100% slow twitch. But based on your theory of if you're sedentary, shouldn't they have some 2Xs? Because they're pretty sedentary, aren't they?
No, I think that there's a different evolutionary path for all these animals. And what we have done is, starting from an ape, which has like usually these hominids have like 1/3 of each type. We are just moving too slow, but every other animal has its own evolutionary path, and the sloth, probably because it doesn't have any natural enemies, they have only slow fibers, and they have their biggest challenge in evolution is not to escape from predators, but to be energetically efficient. So they move very slowly. They can escape a movement detector and so on. They're really slow, and that is something that is also almost forgotten in physiology. But slow fibers are much more efficient to generate a certain degree of force, especially in isometric contractions, so in contractions that are, not just like holding tone of muscles. This is something that slow fibers do much more efficiently. It means it costs less ATP to produce a certain amount of force for a certain amount of time. So that's probably why these sloth, they have such a predominant slow fiber type. But this is the only one we could find that is slower than humans. All the others are faster than humans.
Yeah, they went like boom. Yes, I saw your figure, and yet we were sort of 50-50, and then the sloth is just bang, 100%. Yeah. And there's no variation. Yeah, I was thinking about, it's really interesting how you mentioned earlier the paper, I think it's Anderson, where they rapid switching off of the 2Xs after doing weight training. So like within 4 days, it was all gone. Yeah. What about the other way? So if you go from like, do people know if you're like I guess it doesn't matter if you're weight trained, if you're endurance trained or whatever, you're saying you lose your 2X. But what about if you stop? So you said sedentary people tend to have some 2Xs. If you stop, do you go back, get some 2Xs again?
There is some suggestion in the literature that when you do tapering, then the type 2Xs come back, and that there's even some kind of overshoot of type 2X fibers in your taper, and that gives you this extra kick after tapering. But I'm not sure. this isn't really a consistent finding. It could also be just an accidental finding, but because I don't really think that in these elite athletes that type 2X fibers really have a meaning altogether. But if you go to really sedentary people, we do see that people with a lot of these type 2X fibers, which is like, in my opinion, a failure to suppress the fiber type that we don't longer need as humans. So if there's like a resurrection of these type 2X fibers, that this has metabolic consequences in a way that we see a lot of type 2X fibers in people that are with type 2 diabetes, and also insulin resistant relatives of people with type 2 diabetes. We see a lot of these type 2X fibers. So I think it could even have a metabolic consequence or a penalty almost, to have this fiber type re-emerge.
Oh, wow. Okay. And I guess is that partly because type 2 diabetes is partly genetic. So do you think they may pass that on to their? So this type 2X we're meant to be suppressing and getting rid of. You think if someone's sedentary, they could pass that on? I guess that's epigenetics. So
yeah, we don't really know, but it could be that from a genetic point of view, there's well, like we know with athletes, the fast versus slow. I'm not even saying the type 2X, but even the fast versus slow fiber type, we see that well, there's quite some literature. It's since the last 40 years, I think, showing that the chance to become insulin resistant is higher when you have a lot of fast fibers. I'm not even talking 2X, just fast fibers. Yeah. There was a recent study by a group of Abram Katz in Stockholm who measured fiber type in a non-invasive way. That's also something we can talk about, the relaxation time of muscles. And then they saw that people who are identified based on a fast relaxation time, so a fast topology, that they have much poorer insulin sensitivity, or they have some early insulin resistance almost. So I really think that is an important aspect of trying to predict who is prone to metabolic disease. It's probably not only insulin resistance, also obesity and cardiovascular disease. That part of this reason why some are more prone than others is because they have an innate or inherent fast topology and muscle. It's a project that we are gonna start next year with some new people in my lab, and we're gonna try to have a big study rolling on whether that is indeed the case, that muscle topology can be a predictive factor in the chance to get the metabolic disease.
So maybe so that sort of fits, I guess, with what you were saying earlier, evolutionary wise, that not just getting rid of the 2Xs, but across time we should be getting rid of the 2As as well, right? Because if you're saying that we're built to be slow, to wear down animals with persistence hunting, and then also we're going to die off by getting diabetes and things if we have more fast fibers as well. So you're saying across time we should end up, I guess we don't want to be like a sloth, but having more slow twitch, it's protective in terms of being able to hunt and not get things like diabetes, perhaps. Yeah,
the thing is that we will probably, I don't think that the type 2A will also be suppressed. I think there's always this two roles that you need to have. You need for some actions you really need power, even just to survive, and for others you need endurance. So we always gonna have both. But the fact that we get insulin resistant and diabetes and obesity and so on is not really a driving force for evolution. It usually happens after we reproduce. So we're not going to select this out.
Sure.
It's more like how I see it is like if you're born with a lot of slow fibers, and we know slow fibers are insulin sensitive, much more insulin sensitive than fast fibers. So that's the reason why we people with a lot of slow fibers can handle glycemia much better. Is that that it probably means that if you're lucky to be born with slow fibers, you could probably, well, do without exercise and still be healthy. And if you have a lot of fast fibers, you probably need to exercise and take care of your diet to stay healthy. But that is more of a provocative idea at the moment.
Yeah, it makes sense that we need both fast and slow, because I guess if you're trying to wear down an animal, you want slow. But if something does come at you, whether it's another human or a cheetah or something, I know cheetah, you got to be only like 20 meters away from the tree, but you still want to be able to get there quickly. And fast fibers help you to do that. Now, we've talked about invasive ways of measuring muscle fiber type with biopsies and things. But I know you've been looking at, and you mentioned the ABE-CAT study. Why don't you talk to us a bit about some non-invasive ways of measuring it? So I remember when you first sent me some dot points, or we sent each other some dot points. I remember the old classic sort of 30-second sprint. So you do a Wingate, in the lab, and you'd look at the ones that had the highest force, but then they would fatigue the most. That would give you an idea that they had a lot of fast fibers. Now, we weren't thinking about 2A and 2X, just fast. And the ones that didn't produce as much force and then didn't fatigue as much were slow. Yeah, so that's, I guess, one way. Why don't you tell us, is that a good way of measuring it still? And what are the other ways you can try and work it out?
Yeah, well, I think the reason why we eventually want it is because these biopsies just invasive and so on. It's never gonna find a real way to the elite sports scene, so to say, or talent identification and so on. So we need to go for non-invasive proxies or estimators of this. Well, or we call it muscle topology. It's very fast or slow. There's been measures relating to relaxation time, where you have electrical stimulation of a muscle, and then you measure the time you need to produce force, and then it's actually the twitch, so to say, like fast twitch, slow twitch. You measure the slow twitch or the fast twitch in an electrically stimulated muscle. That's something you can do in humans. It requires a little bit of technique, and that is a very sensitive way to do it. If you go to other techniques that are more functional, like sprinting, for example, you could say, well, I just look at my stopwatch and say, I can recognize a fast athlete. I just see if someone is athlete, or I can measure it if it's athlete. But there's so many factors that influence running speed. So if simply measuring running speed is not a good way to measure the topology of an athlete. So the best thing, because and there's also other techniques like jumping, like these Bosco jumps that used to be popular to measure muscle topology. So because if you have a lot of fast fibers, you are explosive, and you can have more height in your vertical jumps.
It's like a vertical jump.
Yeah, that is something that reasonably correlates with muscle topology. But for all these functional measures, a lot is dependent on whether you're used to do it, right? If you're trained, if you have a good technique of jumping and so on. So I can work on your jumping technique with plyometric training and so on, and make you jump 5 centimeters more in a couple of months. That is showing that it has its limitations in non-invasive measurements. So what we wanted to do is develop a technique that is independent of any form of skill or exercise or fatigue. Just measure the characteristic of a muscle at rest. You can sleep, or you can be trained, or you can be tired. Doesn't matter. You just measure that. And the way we did it is there's a non-invasive way to measure the chemical composition of your muscle with an MRI scanner. So MRI is just a magnetic scanner that you usually use for imaging, but you can also measure the chemical composition of your tissues. It's called MR spectroscopy. So we use that to measure a molecule called carnosine in muscle, and that is a molecule that is typically present in fast fibers and much less in slow fibers. And we thought, well, maybe if someone has a lot of carnosine in his muscle, which we measure non-invasively just at rest in a scanner, that means that he has a lot of fast fibers. So we compared it to the biopsy technique. We've done 3 different studies now to validate that, and we could indeed see in all those studies, men and women, that the non-invasive estimation of carnosine is a good estimate of your muscle fiber type composition. You can do it at a relatively young age, so you could even, I think, use it for talent identification.
So just out of interest, so you did, I guess, 4 biopsies or something, yeah, to get your fiber type, and that's probably the gold standard. And then you do the carnosine with the MRI. And how close was it? How are we talking 5 or 10% or?
Well, so we did it in 3 different occasions. So the correlation is our square of 0.50. So 50% of the variation was explained. But we know that the biopsy in itself, or the gold standard, is also so variable. So what we did then is, instead of worrying too much about whether it exactly correlates with a biopsy technique, which is variable as well, to do the same studies that have been done for the biopsies. So we checked whether elite athletes in short distances in athletics would have more carnosine than endurance, and that was indeed the case. It's about twice more, and we've done that for cycling as well, and we couldn't see it on swimming, and that is another story why we couldn't really see it. Also in the biopsy studies, people cannot really see that short distance swimming requires more fast fibers than long distance swimming, and we also could not see that in the carnosine measurement. But for most other sports, we could confirm that it is a good non-invasive proxy of muscle topology.
That's interesting. I wonder if the swimmers is partly because they all, maybe not all, but a lot of them do so many hours a day training, even some of the sprinters. Maybe they...
I think it's because they never perform at near maximum power requirements, because when you go through the water, I mean, it's a very submaximal effort, because if you do it too fast, you just slip away through the water. So the maximum power in like 1500 or 2000 watts that you see on a bike in track cycling, or you see it in a 100-meter sprint and so on, in swimming, it's only a fraction of those maximal powers that you use even in a sprint, because you cannot, I mean, your capacity to produce power is much higher than you need to just move water away.
That's true. And I often think I'm picturing Ian Thorpe, who is our famous swimmer way back in the 2000 Olympics. He'd be sprinting it, but he'd have these slow, beautiful, strokes. And I'm picturing that, and I'm thinking, yeah, that looks like if he was doing, weights or something, he'd probably be generating a lot more power each stroke.
Yeah. So that was one reason that I went to Australia and the Gold Coast in the group of Claire Meynihan, specifically because it's the highest concentration of top swimmers you could find in the world, or actually training on the Gold Coast. And then we did the scans on all these swimmers, and we still couldn't see it in these absolute world-class swimmers. So then we knew for sure that it's if we can't find it there, it's probably not an issue in swimming as much as it is in running and cycling.
I guess it's so much technique as well. Yeah, running and cycling is just not as much technique. Okay. Now I'm still thinking about that 30-second sprint. So how does that go when you correlate that? Because I remember, was it Thornsensen or something? They did 30-second sprint. They did 30 contractions on the isokinetic dynamometer, the sort of, I don't know if people know that Cybex, Kincom sort of things. And they correlated that with fast and slow twitch fibers. How does that work out? Because that seems like an easy way to do a measurement.
Yeah, it's true. So before we set up a big study to compare all the different techniques that had been proposed, and we first did a literature search, and we came up to over 100 studies, including the one you mentioned, that have a kind of functional measure related to muscle fiber type composition. So then we did a study on 40 people, 20 women, many biopsies, and then did all these techniques. And one technique test we did was the 30-second Wingate test. And there's two elements of that really correlate well with muscle topology. And one is the slope of the fatigue, which makes sense, right? So if you start low and you keep your level quite good, then it means you're a slow twitcher. And if you start high and you have a steep decline, it means that you're fast twitcher. So that makes that is indeed something we could confirm. But the second one, which came out really clear, was lactate levels after such a 30-second all out, and you have to wait a couple of minutes. So we saw after the value you get, like after 3 minutes, correlates really well with muscle topology. So there are a number of techniques you could use, and I think what we have, I mean, we haven't published a study yet, but one thing that we want to promote is that there's many different ways to sample the muscle topology of your athletes, but you have to be aware that some are trainable. So then you should make sure that you do it in a homogeneously trained population. Some are sensitive to disturbing factors like nutrition and so on. And so it's better probably to take different aspects of muscle topology. So one that measures the fatigability, and one that measures the power, and the other that measures like the contractile properties or the relaxation time and so on. And if you make a combination of a few of those tests, you can get a reasonable good idea of the topology of your athletes. And that's when it really becomes interesting, because that's what we've forgotten to do. In the 70s, and I'm glad you had to talk with Dave Costill, and I'm sure if Bengt Saltin would be alive, he would also have been on your podcast. And they discovered so many important aspects of muscle fiber type composition in sports, and we don't use it at the moment. And part of it is because we kind of gave up on how to measure it.
Yeah, so I mean, I'm just wondering, so is your carnosine, I mean, the gold standard is biopsying four biopsies or something. The carnosine, is that better than, say, a 30? I'm still thinking, like, so if someone's listening now and saying, I want to work out my fiber type, it's probably, you'd have to find someone that knew how to measure carnosine and go and have an MRI done. Is a 30-second sprint or something like that, is it accurate enough to get a bit of an idea, and then which I want to expand on, to then apply it to sports, which you just touched on, which David Costill and Bengt Saltin were doing back in the day?
Yeah, well, one reason why we did that study is we've done almost 15 years of work now on this carnosine, which we think as a research tool and in our hands, because we're used to do it, it's a very convenient way to do these measurements in populations that are very difficult to access, and in sample sizes that are very difficult to get. Like, for example, we did this analysis in football players in Manchester United. There's no way you could ever have them do these. They don't even do VO2 max testing or whatever. They're very difficult to get. But MR scanning, I mean, we just said, okay, have your music on and sleep for 20 minutes, and we could have them do that, and in that way we could actually discover things we hadn't discovered before. Like the hamstring strain injury, which seems to be like a thing that mainly happens in fast twitchers. So I think that there's this technique of carnosine measurement in MR spectroscopy is a really good research tool. It's used in a couple of places now, but to make it a broadly applicable technique, we want even more simple tests, and that's why we've done these additional comparisons now. Just make sure that you choose the right ones. I mean, we did a survey with with coaches and sports science staff on what they think about muscle fiber types. Do they think it's important? For what would they use it, and so on. More than 500 respondents. So Eileen Levens, a postdoc in my lab, has done this survey. And sometimes athletes or coaches say, well, what I use is reaction time to measure. But that is not a good measure of muscle typology. No. That is something different, right? Or so you have to be aware that some techniques are really good. And for example, a 30-meter sprint is dependent on so many aspects: the morphology, the size of your limbs, the muscularity, and so on, the running technique. So that is something you should probably not use. But some other things, like biking for 30 seconds all out, is a very good one. So you just have to make sure that you don't do anything. I mean, characterizing a fast topology player may seem easy, and every spectator in a football stadium knows exactly who is the fast players in the team, but it's a little bit more complex than that.
Okay, so if we pick up, you were saying things that David Costor and things people were thinking about 50 years ago or so. Where do you think we should go? So you touched on, how these things can be used. So applying the muscle fiber types. So you touched on a paper you mentioned where you found people that had more fast twitch based on your muscle carnosine had 5.3-fold higher risk of sustaining hamstring strains. Yeah. So that's something you can do to look at the risk of a particular injury, I guess, and then try and train them to minimize that. What are some ways you could use the fiber types then?
Yeah. So as far as I recall, the very first idea in the time of Costil and Saltine and so on to use fiber types is to tell an idea, to say, well, you're a fast twitcher, we see that, why not choose this discipline and you will become good at it? And in East Germany and so on, they did biopsies of 12-year-old kids and so on in that period. Okay. So it was a little bit sketchy, but what we kind of discovered in the last 10 years by using this non-invasive measurement to actually do it in large groups of really professional and elite athletes is that we came to some new applications that were not in the original plan or the original promise of muscle topology in the 70s. And one is indeed the hamstring injury risk. It's very difficult to measure. So what we did is over 100 professional football players. We followed them for 3 seasons, but we had this muscle topology measurement on all of them. And then we had enough hamstring strains, which is the most common injury in football players, to actually have a statistical power to see whether it's different in fast twitch versus slow twitch players. And then we could see indeed there was a 5-fold difference, 5-fold higher risk of hamstring strains in fast twitchers and slow twitch. We saw that in the Belgian competition, 4 teams, and we also saw it in the Premier League competition. So it's something that you can only discover, I think, when you have high enough power and non-invasive techniques. Why that is probably because fast twitchers, they sustain more fatigue. Freek van Kastelen, a PhD student in my lab, has just published a paper last week on the match profile. So he had the GPS data of football players in fast twitchers and slow twitchers. And maybe that's interesting to say as well. In a football team, you have everything. You have the marathon runner, you have the sprinter in your team. So it's a very heterogeneous population. And what we see is the decline in high intensity actions is steeper. From the first half to the second half is steeper in fast twitchers. So they have more fatigue. It makes all the sense, of course. And we know that fatigue is a risk factor for hamstring strains. So I think now we kind of published the explanation afterwards. We first said that there's more injuries, and then we said, yeah, it's because there's more fatigue. So how can you avoid that? That is to take better care of fast twitchers, to give them more recovery, to have the match intervals longer, to just individualize training recovery cycles to the fast twitchers in more respectful way, so to say.
Maybe also I had Christian Thorborg on talking about physiotherapy in sport, and he was saying how eccentric contractions can reduce your risk of hamstring strains. Contracting the hamstrings eccentrically. So maybe you could focus those players and make sure they do that or something like that. I think it's to do with fascicle length, which also seemed to be a risk factor. So I'm certainly not saying it's the only one, but it's a new one we could add to the whole scheme, and something you can work on. You can actually make sure that the fatigue sensitive players are more taken care of, so to say. We did a study in colleagues in Australia and Gold Coast, having an overload of training in runners. So the normal training load is like 100%. If you put in 100%, and then you kind of give an overload of +10, +20, +30% running volume, then what we could see is that slow twitchers actually cope with that. They swallow that. You just add 30% of your mileage, and they're okay with that, and they improve. And we didn't have any fast twitchers in that group because they were middle distance and endurance runners. But if you have the intermediate topology runners, they could not cope with it. They started to have signs of overtraining. So that really means that muscle topology for sports practice, I think one of the most important things is that it allows you to understand your athletes and to individualize training recovery cycles based on these individual differences. Okay, so that fits with your saying the fast fibers are more fatigable. So the soccer players and the endurance, the soccer players, as they get more fatigue, they're more likely to do a hamstring strain. And then the endurance runners, when you increase the load, they've got more fibers that become fatigable, so they can't handle it as well as the ones that have a lot of slow fibers. So that's really interesting. So you can really apply it. I was thinking, my first thought with the soccer players, well, I should say football, I guess. The football players, in Australia, football can be rugby league, rugby union, Australian rules football, and soccer. So we have to say soccer.
I don't get it.
You get it. Good. My first thought, I guess, was with the football players, that the ones that had the more fast twitch would be faster, and they'd have more forces that, they'd have to be slowing their leg, because, obviously you're lifting your leg and your lower leg's going forward and you've got to slow it. And that's what the hamstring's doing. I was assuming that the faster ones would be getting more injuries and they'd have more fast fibers. But you're saying it's the fatigue. So based on that, do people have hamstring strains more in the second half of the game than the first half as they become more fatigued?
Yeah, so it's even in the first half, the last quarter, they have the most hamstring strains, and then it goes even higher in the second half, and it's the highest is at the last 15 minutes of the last half. So that is what had been shown. So fatigue is certainly a factor, but maybe interesting to say, and that's also something that people don't know about fiber types, is that fast fibers are not so robust, are not so endurable. It's like a Formula One car. I mean, it's really everything is made in a very light and very for optimizing speed. So the Z discs are much smaller, thinner, and the whole machinery of fast fibers is like it's more brittle. It's not so robust, and that's maybe also a reason. If you just put a mechanical stretch on a fast fiber, is more likely to break than on a slow fiber. So it's probably also that factor, and that is something you cannot change with individualizing training recovery sites. So maybe there's other factors as well leading to more injuries in fast fibers.
Okay, just other things. Just thinking about other ways to apply your fiber type topology that you've worked out. So we've talked about hamstring strains, and what was the other one we just talked about? I've forgotten already.
The training recovery cycles and
the
overtraining.
And then what was the one? Overtraining. That's right. And what was the one you had? Tapering. Something about tapering as well.
Yeah, but there's not a lot of information on that. But what we could see in that same study. With overtraining, we could also see if when they had a week of taper that in that period the slow twitchers could really rebound quite well their performance, but it's more a research idea. I don't think at the moment we can say there's sufficient information to say that to people that the taper in slow twitchers can be shorter. That's my hypothesis, but we don't have that direct evidence yet.
Okay. I guess that would fit with them being able to recover quicker as well, right?
Mm-hmm. Yeah. You must realize that a slow fiber cannot fatigue. It's maybe strange to say, but it cannot go into acidosis. I have a colleague in Poland who does electrical stimulation of single motor units. So what you do is you dissect in a live rat in the spine. You dissect one motor unit, motor neuron, and you electrically stimulate it, and then you look how much you can continue the contractions if they don't know in advance, but if they hit the slow motor unit and a slow fiber and they contract it contracts all the time at the same force. You can stimulate it for hours and hours and it never fatigues. I mean, you could go on for days probably, and slow fiber you cannot fatigue. So we have to realize that's exactly what we do. We have these slow fibers who are active for hours a day. So they are so different, fast and slow fibers. And that we must realize that if you have an athlete in front of you that has a lot of slow fibers or a lot of fast fibers, that makes a lot of difference for many things. So we have this hypothesis that it also impacts nutrition. I think that some nutritional supplements only work for fast twitches and other supplements only for slow twitches. That's another project that we recently started in our lab. So I think the applications are in many directions. And when we asked these coaches, for what would you use it? Why do you think muscle topology is important? Over 20% said, well, to individualize nutrition. And we were like, what? There's no science on that. Oh yeah. To individualize nutrition. So I think the field is ahead of us on that.
That's interesting.
On that part.
So what would they say? What would they say? And what are your? I know the science isn't there yet, but what are they saying? Like, no, why don't you tell me? What are they saying?
Yeah, I think it's in two directions. It could be the fueling. We know, for example, that fast fibers, you can increase the oxidative capacity. You can make the mitochondria, but you can never make them good in burning fat. That is something that's like almost exclusively done in the slow fibers. So that is certainly one part. The macronutrient combustion, the fueling, I think is one direction, and the other is the supplements. For example, the dietary nitrate supplementation. You had Andy Jones in your podcast series as well. It was a Swedish group who showed that only fast fibers have a benefit from nitrate supplementation. So that means that if you have a fast topology, you will probably benefit from beetroot juice, and if you're slow topology, you will not. So I think also for that, to have these nutritional supplements, effectivity, to explain why some people are responders and non-responders. I think quite a bit will be explainable by muscle topology, I think. But all that is still to be done.
You know what? Sorry for being Australia-centric, because I mentioned Ian Thorpe earlier, Australian swimmer. When you talked about slow fibers, you can't fatigue them. It reminded me of, we had this great marathon runner, Robert De Castella, in the 80s. Anyway, I remember his 400. The idea is when they tested him at the Australian Institute of Sport, they couldn't get his lactate up. Just couldn't, do a sprint, whatever, just no lactate. And then it makes sense because I remember at one stage at the end of this marathon, I think it was the Rotterdam Marathon, he did 60 seconds for the last 400 meters, when his best ever, like fresh on a track for 400 meters, is 57 seconds. So it's just like one speed, and can't produce lactate. So just same speed the whole way, probably.
Yeah. And also the other way around, you have those stories of these extreme fast twitch athletes who have never run more than 400 meters in their life, right? it's amazing. And if you would have them go all out on a 400 meter, they would probably have to recover for a week or something like that. So it's a true thing. And I was talking about Homo sapiens being slow twitch. That's true. But we must realize that we still have a big variability, a big range. And that is something that I think is, it's also a hypothesis. I'm throwing a lot of hypotheses here. I think the variability in slow twitch, fast twitch within a community has an advantage as well. I don't know if you know about chronotypes, but there's people who are early birds and night owls. And I think having the combination in your group or your small population is very valuable, because then you're always protected from predators during the night, because there's always some late night owls still awake, and then they are replaced by the early birds 1 hour later. And I think it's also the case for fast twitch, slow twitch. A tribe would probably be stronger if it had an endurance runner and a sprinter and a tree climber in the community to take all the specializations. And maybe that is a reason why in humans there's still a big range, there's still a big variability, and we exploit it, and we enjoy it in sports and in Olympic Games. But maybe it's a deliberate choice and evolutionary strategy to be broad ranged.
Definitely, you'd know when you do these pracs in your undergrad classes and things, you'll get people doing 30-second sprints. I know I keep talking about that, the wind gate. So just so people are clear, so 30-second sprint on a bike is the wind gate is where you go as fast as you can from the very first pedal, so you don't pace yourself. You go as fast as you can, and then you hold on for dear life for the 30 seconds. It's probably the longest 30 seconds you've ever had to endure. Yeah. But the funny thing is, you'll see, and you're talking about this variability. You'll just get a bunch of students, one from each group does a 30-second sprint. One person, massive force, and then they die like crazy, and you've got to watch them. They'll go to the bathroom. You got to get someone go with them because they can faint in the bathroom, hit their head, whatever. And then you've got the other one that generates nothing, fatigues like nothing, and then 2 minutes later they're like, "Oh, can I have another go?" And the person that's with the fast twitch person's throwing up, and what do you mean? They're fainting and whatever. So yeah, very, very different. And just so hopefully people would get this by now. But the person that does the high force and fatigues a lot is going to have more fast fibers than the person that's
there was
one that doesn't fatigue much.
Yeah, there was one study in our lab from Eileen Levens, who is now postdoc. She did 3 wind gates. So I mean, if you do 1 wind gate, you regret it after 5 seconds that you ever started at. So that 30 seconds all out, and then you give them 4 minutes of rest and do another wind gate, and then 4 minutes of rest and do another wind gate. So majority would then vomit and everything. It passed ethical committee, and then we did that in a group of fast twitchers and slow twitchers. We had defined beforehand. We had we left the middle part out, and we just did the scan on this is a group of fast twitchers and slow twitchers. We did the 3 wind gates, and then we also looked. We indeed saw these fatigue profiles to be very different. But then we looked how much time would it take to recover, and the slow twitchers, they recovered within 30 minutes. And the way we measured the recovery was the maximal voluntary contraction of the knee extensors, so just an isometric knee extension, and that was back to normal within 30 minutes. But the fast twitches, and we only measured for 5 hours, so we don't know what happened after that. But they were still not recovered to baseline within 5 hours. So it could probably take many more hours or maybe days for them to really recover. And they did exactly the same total power. So it was distributed differently, but they did exactly the same amount of work. It was heavy for all of them, and they didn't like it, all of them. But the way they differed in their recovery was so massively different. And as a coach, we don't realize how much difference there can be in how, when you ask something, run a couple of rounds, and I mean that just can come in so differently from a physiological level, from a physiological point of view, can come in and kick in so differently in athletes. And you have to take care of this individual variability and learn to understand that these are not weak athletes. These are just athletes that have a very different physiology. Just the way their muscles are composed, that's basically it.
That's interesting, because I remember, again, talking about some sports people don't know about, but rugby union. So during high school and then early 20s, I played rugby union. And back then, because I'm 61 now, so, early 20s, everyone trained the same. Sorry, the coach would get everyone do, 3 laps of the oval, and then everyone do these sprints and everyone do this. It's like, and then later on, people start going, well, hang on a minute, they're very different. The fullback stands there, does nothing, and then sprints, and then the forwards are like running around all day, and just like in soccer, I guess. So then we got to the point where we start training them. Sorry, we start training them differently. But now you're saying, I guess, that we've got to get to the point that even within those, so you've got the forwards and the backs or whatever, even within those, you're going to have some that are going to have more fast twitch and they're slow twitch, and you've got to think about their recovery will be different as well.
Yeah, it's in some sports it is the case that different positions require different fiber typology, but not in soccer. We don't see that. We've done it in many teams now, and there is not a clear pattern. We've done 150 professional players. So if there is a difference, it's probably a very small difference. If you need many more hundreds of players to prove your point, it's probably only going to be a marginal difference. So there's two things. You don't need to be of a certain type to be a professional football player, and the type of your muscle topology is not going to define where you play in the team.
Okay.
So it's other factors. It can be morphologically, I mean, length and so on, muscle mass, this kind of, but not so much muscle topology. Yeah, but in other sports it is, of course. I mean, in rugby, it's quite clear that if you run a 100-meter dash in 11 seconds along the line, that's not going to be for everyone, right?
All right, so I think we've talked a lot about fiber types. What I'd like to do is ask, is there anything else you're doing in the lab that you're excited about? So I know you sent me something through. So why don't we talk about that a little bit? You're talking about the histamines and
yeah. So I really focused my research on the muscle fibers, the myofibers. And a colleague of mine, Katrien de Bok, she's a good friend, she was also on your podcast. She always said, yeah, Wim, I'm not so interested in muscle fibers. I said, what? Muscle, skeletal muscle. I mean, the majority is muscle fibers. That's what, no, I'm interested in muscle physiology, but not muscle fibers. So she's interested in all the other cells that are in a muscle. And now I'm also starting to get really interested in that. Because there's so many, we call them mononuclear cells, so the multinuclear cells, or the myofibers that are like grown together cells in a long line. But the mononuclear cells, this is just a cell with one nucleus. You have all kind of immune cells, it's called FAPs, and on all these different cells. We don't know nothing about them in muscle, and they seem to be really important. And what we did is and this study is still ongoing. It's Thibaut van der Stede, who was a PhD student in my lab. He looked at transcriptomic level, which means if you have an exercise and you look in a muscle, how many genes are upregulated or downregulated by exercise, which seems to be quite important because that's probably going to predict what's going to happen on the long term in training adaptation. We see that the majority of these changes in upregulated or downregulated genes are not in muscle fibers, but in other cells, in immune cells and so on. And that is quite surprising, I think, to us, because we always think about how does a muscle adapt to exercise. We see the mTOR pathway going up and AMPK going up and so on. Forget about the myofibers. What we've done is we suppressed the histamine signal during exercise. So histamine is something that is liberated by mast cells. It's a signal molecule, and it's well known for allergy medications. If you have allergy symptoms because you have too much histamine, if you block the histamine, then you don't have these symptoms. If we give antihistamine during exercise, then you don't adapt to exercise. You don't have a training effect. And we did it in a 6-week training study. We gave them antihistamines with each training session, and hardly anything happened. They didn't get more mitochondria. They didn't get more capillaries. So all the training effects or the health promoting effects were gone. And the really interesting part is muscle fibers, they don't have histamine receptors and they don't produce histamine. So the whole thing was blocked. The training adaptation was blocked, and the myofibers had nothing to do with that because they don't even have a histamine receptor. So that was the first study. We did it 2 years ago, and now we started looking into all those other fibers, or all these other cells, and they seem to be the key players in training adaptation. We've never thought about even the slightest idea that it would not be the muscle fibers, but it is mainly the other fibers, having all the other cells, all kind of signals going back and forth and so on. So I think a main thing that we will see in the next years is a discovery of all the roles that these other cells have in muscle. I mean,
wow.
We now just saw that there's mast cells in muscle. We never knew that there was mast cells in muscle, and they seem to be just part of a very normal training adaptation signal. It's not pathology, it's not infection, it's not muscle damage. It's just you exercise a muscle, and mast cells, immune cells start a whole cascade of signaling that actually leads in the end to more mitochondria, more capillaries, and so on. So that is, for me, an eye-opener and a game changer in the field. And luckily, we can now do these studies with single-cell transcriptomics and single-cell proteomics and so on. So this is really going to give us a whole new perspective of what exercise physiology is all about.
Wow. Okay. Well, I'm trying to get my head around this. So you're saying that when you exercise, it's not the muscle fibers, because even though they're contracting and they're getting these signals and things that are going on, you're saying none of that matters. It's what's happening to these mast cells that are producing histamines. And even though, but then you said the muscle fibers don't have histamine receptors. So how does the muscle fiber even know that the mast cell is producing histamine then?
Yeah, histamine is just a first signal from one mononuclear cell to another, and in the end, it will also come, of course, to the muscle fibers. I mean, I'm putting it a little bit black and white. We also saw a lot of transcriptomic changes in the muscle fibers, but these were proportionally only a minority of all the changes. I don't know if you're aware of the Metamax studies. No. So these are just studies where you have a transcriptomic response in muscle, but that is bulk muscle. And that is, it's a really nice overview of all the transcriptomic studies all combined in one like meta-analysis and tool you can use. Just realize that most of these changes and up and down regulation is in other cells. There's also some in muscle directly, but the signal isn't probably starting in muscle, but it's ending in muscle. It's all other cells that are doing
this might be
things we don't realize. Yeah,
this might be out there somehow. But I had Mark Tarnopolsky on, and he was talking about all these different mitochondrial diseases and glycolysis diseases. So people that don't have phosphofructokinase, so they can't really use glycolysis. So people know glucose and glycogen go into lactate. McArdle's disease, where they can't break down glycogens, they don't produce lactate, whatever. But he basically finds that all of these different diseases can be improved by exercise if you train them. So if they can't use glycolysis, then if you train them for their fat oxidation, they'll improve. Or if they don't have fat oxidation enzymes, if you train their glycolytic or their carbohydrate pathways, they can improve. So it's hard to see, because that looks like it's going straight for the muscle fiber defect. And it's, and they do get these training responses. It's hard to see how, you know what I mean?
Yeah, but still it doesn't exclude that also in these patients, it's the mononuclear cells that set up a lot of the signaling and start a lot of the adaptation. It's
what I mean
just
is the adaptation is well, I guess that could still work, because the adaptation is okay. So if you don't have phosphofructokinase, you can't produce lactate. You train for fat, and you get better. But it could be that's still just the signal is just to train fat. It doesn't matter that you've got that problem. Yeah, I get what you mean. Wow. Okay. Well, I'm gonna have to look up the. So you've done that study. Have other people followed up on that with the? So you're saying if you take antihistamines, it's not a good idea if you want to adapt to training. So are these really high levels? Is it the level that someone's taking just for hay fever? Would that be enough to stop your training adaptations?
Yeah. So we've done studies with high doses of both H1 and H2. So that's two different types of histamine receptors. To completely block all the histamine signaling. Now we are following up on a study where we do only the H1 receptor blockade, the one that is used for allergy medication, and in the dose that is used by people normally. And what we do is we do a resistance training, and with every resistance training bout, we just give them the normal allergy dose and medication H1 just before the exercise or to the resistance training bout to see whether that would also affect. So that's, of course, the question that people ask us a lot. I mean, this study has been really picked up in the popular media for that reason, of course. But I cannot answer that whether it's really problematic for people taking antihistamines. But it seems to be the case that we underestimate how much damage you can do with drugs. We of course always realize that the side effects, but in the case of histamine, there was histamine signaling hundreds of million years ago, even in single cell organisms, long before there was mast cells. So histamine is a really old molecule that is used all over the body for all kind of signaling. And if we use antihistamines just for one reason, to get rid of your symptoms of an allergy, just realize that all the histamine signaling in the rest of your body is gone as well. And in the case of muscle, it seems to be quite a problematic impact. It's also for glycogen resynthesis, and that's something that would be interesting for you. If we have H1 blockade, not H2, but if we have H1 blockade, the muscle glycogen resynthesis within 3 hours after exercise was completely blunted by a H1 receptor antagonist. So the glycogen resynthesis in the first 3 hours goes up quite a bit after exercise. It did not go up in a antihistamine H1 receptor blockade.
H1's the normal one that people take with their antihistamine medication.
So it's not only training adaptation, it's probably also simple recovery from exercise. So that's go ahead.
So I know Ilva Halston said just a couple of weeks ago that we were talking about post-exercise hypotension. So after exercise, your blood pressure drops. And she was saying you could prevent that by giving an antihistamine.
Well, it's along the line of this. I mean, and Ulva is also a co-promoter of the PhD student who is working now on this antihistamines. But the one who started this type of research on Halliwell in Oregon, he indeed first investigated antihistamine or histamine signals because he thought that was the reason for the post-exercise hypotension that you can get. But then he also followed up with a study where he saw that the whole muscle transcriptome was blunted with antihistamines. And that's the part where we picked up and not so much the cardiovascular implications or the hypertension or whatever.
Wow. So just to summarize, you're saying that therefore that antihistamines, but again, you said you don't really know yet if it's just the doses that people take with they've got itchy eyes and runny nose and whatever, but potentially it's reducing your normal sort of blood flow after exercise, which is why the blood pressure drops. You have a greater blood flow. It's maybe reducing your muscle glycogen resynthesis. It's reducing your muscle turning on all these genes and things during and after exercise. And you're saying more chronically, it can be reducing your training responses. So is that fair to say?
Mm-hmm. Yeah, that's true. And this is only one signaling molecule. And I think there's a lot of others that are also activating. And this whole training is actually, you change the interstitial milieu. You're soaking your muscles and muscle fibers and all the other cells in some kind of exercise bath. And then the whole thing is upside down, and all cells are activated and communicating with each other, and you really create one big soup of exercise signal, and all the cells in there start communicating with each other and adapting, and that's what eventually leads to training adaptation more mitochondria, more capillaries, and so on.
Yeah, and you say messing around with that by, say, an antihistamine may throw it out. Similar discussion we had with, I think it was Katrine de Bock, and also when I had Christian Thorborg on again with physiotherapy, this idea that, oh, you sprain your ankle or you're just a bit sore, so you take these anti-inflammatories. Spraining ankles a bit different, but taking anti-inflammatories, then that's also another thing, right? Because so if you hurt yourself, then the timing can make a difference. Should you just let your body do its own thing? But then it's like, oh, I don't want it to swell up too much, so I'll take an anti-inflammatory. But the timing may be important as well.
Yeah, anti-inflammatory, but also antioxidants. That's also been shown by first Michael Ristov and so on. If you take antioxidants, then also your training response is blunted. So reactive oxygen species probably also play a role in this interplay or the communication between cells in the post-exercise muscle environment, which eventually leads to training adaptation. We see a lot of when we do the antihistamine or the histamine blockade in muscles that have just exercised, we see that a lot of the inflammatory signals are reduced, and also reactive oxygen species signals. So I think there's a lot of players in a very complex interplay, and that is one of the big challenges for the coming years, is to understand this better. And if we understand this better, we will also understand why there's adverse effects of certain medication for exercise response.
Right. All right. So just before we finish up, actually, I was thinking, I didn't ask you about, I like to ask about, are there sex differences? So if we go back to muscle fiber type, are there sex differences? And also with age, do you get, I know we get changes in fiber type with age, but why don't you just tell us a little bit about that?
Yeah. The sex differences are subtle, but they are there. Not so much in the number of fast versus slow fibers, but the fast fibers of men are somewhat bigger than for women, and the slow fibers are not different between men and women. So if you add that up, you actually see that when you have just the muscle weight, that men have about 5% more fast twitch muscle weight than slow twitch muscle weight compared to women. But it's a big range, so it's two overlapping Gaussian curves. There's a lot of overlap, but on the average, you see slightly more fast twitch muscle mass in men. So with age, there's different periods where fiber topology changes. One is in puberty. What we see is the difference between men and women, and fast fibers becoming bigger in men. This is something that happens during puberty. And also the fast topology really increases during puberty. So to say, children are like more oxidative, slow type individuals. You see that if you walk in the mountains with your kids, and they seem not to fatigue. It's amazing. And if they're fatigued, and then they wait for like 1 minute or 2 minutes, and they start playing again. It's amazing their recovery potential. So the real fast twitch profile really kicks in during puberty, and then it goes back again. Then we start going to a more slow twitch profile as we go into advancing age. And when you're really talking with the old age, then you see that actually the motor neurons of the fast fibers die off. Sometimes they are reinnervated by slow motor neurons, but then we're talking at a really advanced state. But even before, when you're 40, 50, and so on, you see that on average that slow twitch profile is taking over again.
Right. Okay. Okay. So how do we not how do we? Okay. So this has been interesting. What I'd like to do at the end is to finish up with some takeaway messages. So we've covered a fair bit of ground today. What would you like people to take away from this?
Mm-hmm. Well, one thing is that the fact that we have been like a bit on the wrong leg with this 3 fiber types, and in the end there was only 2. Part of it is because when we do animal research and animal models, we always think that it's exactly the same in humans. And we have to do this translation as well, and have a lot of attention in when we do discoveries to also see whether it applies to humans and how it applies to humans. And some of these findings are lost in translation. And I sometimes find it a bit frustrating. So to see, I was looking at cell metabolism. That's a top journal in our field. And also last week, all of the papers are in mice. And that's very good, but and they have their value. But we should always question ourselves: can we translate this to humans?
Absolutely.
Is it the same? And also pay attention to that. So that's one thing. I think we should never forget the translational part to humans, and then also further to the field, to sports. I think these fantastic discoveries in the 70s by these giants on muscle fiber types, I feel sometimes a bit embarrassed that it's still not used in sports. We should do better and translate it all the way to the field. So that's one thing. Second is that maybe people don't really realize so much that endurance capacity is really our thing. That's what humans are really made of. That's how we want to profile ourselves in nature. And that is probably one of the reasons why we are getting rid of or losing this extreme fast topology. And then, well, third, the last one I mentioned is these non-muscle cells. I mean, we're going to see a lot of work in the next years on how to understand muscle physiology, fatigue, recovery, adaptation by looking at the other cells, the cells that are not myofibers, but of which we know almost nothing at the moment. So that is exciting times ahead. It's not going to be easy. There's going to be a lot of omics and stuff. But we're going to finally understand in a completely unbiased way what actually is happening in muscles during exercise.
Actually, do you want to just explain when you've said a couple of times unbiased? Do you want to just explain to people if they're not sure? Because when you think of bias at the moment of your echo chambers on Twitter or something, but you're not talking about it in that way.
No.
What?
No. So when I was saying this type 2X fibers don't exist, there was when you look at all the proteins or all the transcripts, and you ask a machine, can you put all the fibers into 2 or 3 categories based on not just one gene or one protein, but on all the genes, the whole package? That's what's called unbiased. Biased way is that we say, well, I think the most important protein in the muscle is myosin heavy chain. That is you already have taken a choice, and you don't know if that one out of 5,000, why would that one be the best one? So you're biased, and the reason we're biased is because someone has started, Brook and Kaiser have started ATPase staining, and that measures the enzyme that's present on myosin heavy chain. And from then on, we didn't think for 50 years. We stopped thinking for 50 years about what would be a good way to classify fibers. And now we just say, well, we just take a blank page. Every protein could be a fiber classifier. Just tell me what you think is in this database. And that's what we call unbiased or unsupervised. And that's the only correct way, so to say, to classify subtypes. And we ended up with 2. So that is, well, there is one thing maybe, and that is also in this paper. Sorry for that. But the slow versus fast classification of fibers is not the most important way to classify fibers or explain differences between fibers. So that is something that came out of our study, but it's still not published. So I won't go into too much detail of that. But what we actually found, it is even more important factors than slow versus fast to describe the differences between fibers. And if you want to know what it is, you have to look up the bioarchive paper and read it.
Oh, you can't just tell us very quickly and then they can look it up?
Okay, so it's ribosomes. Ribosomes is a big thing now. What we saw both in the trans.
Just say what ribosomes are quickly. Yeah.
Ribosomes are small. Mm-hmm. Yes. Ribosomes are small organelles in a cell that translate mRNA into protein. And muscle cells have an awful lot of them because the whole protein turnover in our body is, from a practical point of view, the most protein turnover is in our muscles. So these ribosomes, the small machineries that make protein from mRNA, are really active in muscle fibers. And what we now see, that there's ribosomes that have a number of ribosomal proteins, a certain type of ribosomal proteins, and we have cells that have ribosomes with other types of ribosomal proteins. And we don't know at the moment what the difference is, but we know that if you ask in an unbiased way, tell me what the most important difference is between these 1,000 fibers, then the machine will say us it's the ribosomes. The ribosomes are so much difference between from one end of the spectrum at the other. And then the second most important difference between all those fibers is that some are slow and others are fast. So it's a really surprising finding, and we're still digging into why that is. And it's a collaboration with Atul Deshmukh and Roger Moreno and Ben Stocks, and it's really something we enjoyed over the last half year to discover. Yeah.
Great. Okay. And the important thing is with these things is people don't always think. You said that the paper is what people call a preprint, and you often see on Twitter people will say, "Congratulations." You know, someone will say, "I've got this finding," and they'll say, "Congratulations." And you go, "Hang on a minute, it's a preprint," which means it hasn't been reviewed, it hasn't been accepted, it hasn't been published. It's a preprint, and that's a preprint. I'm not saying it won't come out, but people sometimes forget that, don't you think? Have you noticed that?
Absolutely. So, but I also support the idea that it's valuable to throw the data out, and so people can start working on this. Of course, there is a risk that people would spit out data that are completely invented, but you also can have that with data that are published. So it's not really a guarantee. It's published in a paper, so it's free of faults. But you of course have to take into account that it's still subject to change, to critique, and so on. And then in that case, I think it's worthwhile to look at who is writing that. Is that someone who has already had a good or a valued expertise in the field, or someone that is just coming out of the blue? Then I would be a little bit suspicious on preprints.
I'm not obviously putting down your paper. I haven't looked. I did see the title. I'm just saying that concept sometimes gets lost, because there's one thing, you said Cell Metabolism, a top journal. If a paper's published in that, it's probably likely to be tighter than something that's published in a much lower journal. And here we've got preprints, which you don't even know, hasn't been reviewed, hasn't been accepted, hasn't been published. You don't know what journal it's going to get into. But sometimes on Twitter, people will be like, "Oh, look at this great finding." It's like, no, it's a preprint. So I'm just making a point there that I've been wanting to slip out, and you happen to be on the end of it. Okay.
Absolutely correct.
All right. Well, thank you very much for coming on. But I agree with you. It's good to throw it out there, and you can get feedback, and you can get because it used to be people would be very secretive. They wouldn't even mention, which thankfully has not really happened on the podcast. And you're happy to mention things, but used to be people would be very secretive, you know, they wouldn't want to talk about what they'd found and things. I think people have found more and more that there's more to be gained by actually putting it out there and getting other people's ideas to then, you know, follow up, etc.
Yeah, I just realized that I've thrown all our ideas in the lab in the public now, but that's what I think is the most valuable. We have a lot of ideas, and we want to, well, have people think about it. And I mean, like this two fiber types only idea, it will create some debate and critique. But by doing so, by just saying, well, wait a minute, what if we started the other way around? We say there's only 2 fiber types. Then you can throw mud at it and try to disprove it. And if we can't, I mean, then it's probably correct. But if we never challenge the idea and never be provocative, then we will never challenge the idea that there's probably not 3 fiber types. So I like that debate and throwing no ideas on public.
I was going to say, if no one challenged that the world was flat, we'd still think the world was flat. But some people do think the world's flat. It's making a comeback. Okay.
There's people from all around the globe that think it. Yeah.
Yeah, and as you said, there's a lot of variation within people. Okay. Well, thank you very much for coming on. It's been great, and I'll see you around.
It was a joy. Thanks a lot, Glenn.
Okay. See you, mate. Bye-bye. I hope you enjoyed this podcast. And please like, subscribe, pass it on to your friends and colleagues. Check out the other podcasts. Thanks again.